then these cells die. So if you have a tissue, if you just pile cells in the shape of your heart, then within a few minutes, all the cells that are 200 or more microns away from a surface die because they don't get enough oxygen and nutrients. So this vascularization is also a big, big problem in tissue engineering. And the other thing is that this proper function. So even if you have tissues that, let's say you put the right cells in there so that your body doesn't reject it, and you somehow create ways to supply it with oxygen and nutrients, then you still need to put these cells together in a way that they function like they do in a normal tissue. So let's take the case of your kidney. Your kidney is not just a pack of cells. It's got very controlled and organized structure about how it does its function. Same thing with your heart cells and your heart. So all these organs are very complex and very controlled. So if you just pack the cells together, they don't really do the function that they're supposed to. So these are some general kind of problems in tissue engineering. So, so basically what we need to do to push this field forward is to try to understand the biology better and try to come up with better ways of engineering these systems. So the biology of basically organisms is very complex. So every single one of us in this room has come from a single cell that goes through many rounds of differentiation and division to give rise to the organism that we are now. And this kind of process is very, very controlled. So of course we know that there are genes and proteins in the cells that partially regulates this process, but there's also a lot of interaction between the cells, things that happens outside the cells that regulates this process. So for example, during early stages of embryonic development, the cells that give rise to the organism, which are these cells here, actually talk to each other to a great deal. And based on this communication, they know what they should do. So for us to be able to engineer tissues, we need to be able to understand this communication and try to engineer it in our systems to be able to tell the cells that we want to make the tissues that we want. The same thing is not only happening inside during development, but it's also happening in all of us right now. How we're controlled, how our body operates, to a large extent has to do with how cells communicate with each other. So, for example, we know that our blood keeps on replenishing itself. You don't have the same blood throughout your life. It just continuously replenishes. And we know that a lot of this replenishment and regeneration of the blood happens in the bone marrow. And, so some of you guys may actually have seen a bone marrow and you would think that it looks like a soup of cells. There's not too much control and organization in a bone marrow. You just see a lot of blood. But what actually you have when you look at these cells in a controlled way is lots of structure and organization. And this is starting to become much more appreciated these days compared to even 10, 15 years ago. So these stem cells in the bone marrow, which are the ones that give rise to your blood, are just not floating around, but they're actually localized in very controlled environments. And this microenvironment of these stem cells is very organized so that they interact with other cell types. So they can interact in very controlled way with blood vessel cell types, with bone cells, and this interaction tells the stem cell whether they should remain as it is or whether they should proliferate and make more blood cells. So this is happening in all of us right now. So what we're trying to do, and many other people in the field are trying to do, is to bridge this gap of what happens in the body and what you do with the cells when you take them out and put them in a dish. So in a dish, you can get cells. This is a scanning electron micrograph of a cell. And you can see that the cells can attach and can migrate. And there's a lot of interesting biology that has been done with this. But this is very different from what happens in the body. So one of the things I've been talking about is those very complex processes that happens in the body. So this is an example, a schematic diagram of a functional unit of a liver called the liver lobule. This basically what it is that it does all the things that the liver does, except it's very small, and it's the building block of the liver. So the liver is actually made from repeating patterns of this structure. So you can see that it's very different in the fact that this is three-dimensional, it's vascularized, and the cells in there can communicate with each other in very controlled ways. So we need to bridge this gap. And what my lab and a number of other labs work on is to try to address this difference by combining approaches that are at the interface between biology, engineering, and material science. So here's, imagine if you're a cell, then you can see a number of things in your environment. So you can see soluble factors, these are proteins and hormones and other things. And the cell recognizes these soluble factors, but not just in a very kind of random way. It actually senses the concentrations, and it senses how these molecules are present with other molecules. So I kind of like to think about it as, your proteins, your signaling proteins and hormones are like words, and how it actually forms sentences depends on the combinations of these different proteins and hormones that are present. So, and that makes the cell understand the soluble factors. Cells also contact each other, and that directs the behavior, and they can be inside these extracellular matrix or three-dimensional gel-like materials, and that also signals the cells. And there's a lot of other kinds of signaling processes, like mechanical factors. So one of the things, one of the biggest kind of advances in biology has been that people now realize that it's not just your proteins and hormones that regulates what your body does. The mechanics also regulates it. Of course, if you're like a bodybuilder, you've known that for a long time, that the mechanics is very important, but biologists are starting to figure this out much later. So what we do as engineers is we can take these cells and use a lot of different techniques to regulate what these cells are seeing, and then try to understand what they wind up doing, and try to use it for different applications that I mentioned. So how do we go about doing this? Well, one of the things about cells is that they're very small. So if you want to be able to control the environment of the cell and regulate what a cell does, then you need to start modifying your environment at the same length scales as a cell. So there's a lot of technologies that have been developed to make stuff at very small scale. And the most relevant of those is actually the microfabrication technologies, which are used to make your computer chips and cell phone chips and all these kind of microelectronic stuff. So with those techniques, we know that we can make things that are very, very small. So there's a number of approaches that we and other people have used to try to borrow those techniques and use it for our applications of trying to control what the cells do. So for example, you can take these silicon wafers that have patterns on them. These are the same kind of things as your computer chips, right? So very similar. But instead of going to a clean room or these microfabrication facilities to make them, we want to be able to control how we make them in our own lab. And our own labs are very similar to the labs here, so we don't have people wearing bunny suits and things like that. It's much more dirtier than what you see in Intel commercials, right? So what we want to do is we want to take these kinds of micro-scale structures and use it in our own lab. So we and others have developed these techniques where we take these silicon wafers with patterns on them, we pour material on them that's initially it's it's not solid, it's more gooey-like, and then we pour it on these silicon wafers, and when we pour it on, then it becomes more kind of rubbery, and we can peel it off, and we get structures like this. These are transparent structures made of this material, polydimethyl siloxane, which you don't need to know the name, but this basically, you can then use it to make channels, to mold materials, or to pattern surfaces, all with the goal of trying to control what these cells are seeing. So for today's talk,